Okay, so we're going to switch gears a little bit and we're going to talk about DNA versus RNA. And what's really sort of interesting is that we think that life really began with RNA being our genetic material, right? But then we transitioned to DNA. And so in order to understand why is DNA so much better than RNA, we have to really look at their building blocks and their three-dimensional structures, right? And so we know that there are three really big differences between DNA and RNA. We know about our sugar, right? That we have that 3 prime, it's not a 3 prime, it's the 2 prime. It's the 2 prime that has the hydrogen or the hydroxyl group. Okay, so deoxy has hydrogen, this one has OH. Then DNA can have thymine, whereas RNA has uracil, and we'll talk about why that's super important in a little bit. And then really DNA has some super interesting complex structures that it creates. DNA basically makes one, it makes that double helix, right? But RNA can make some super interesting three-dimensional structures. And one of those three-dimensional structures turn them into ribozymes, which make them enzymes, which make them catalytic molecules. They can actually cleave things. It's really cool. So why is RNA so much less stable than DNA? So if we look at our backbones, right? So here's our RNA, and we have 2 nucleotides here, right? So we're in basically a basic solution, right? A basic solution. And so what we have here is OH minus that can come in and it can actually interact with the OH with the oxygen on this 2 prime sugar, right? And when it does that, this oxygen here undergoes nucleophilic attack on our phosphate. And so what happens there is that it's actually going to break the phosphate backbone. That's part of the phosphate backbone. So it's going to create a break in the phosphodiester bond. So we call that an autocleavage event. And then it actually creates a break in the RNA strand. And this happens all the time. So those free hydroxide ions, right? They deprotonate that hydroxide right there. And so because they're deprotonated, right, then they're going to go and attack that phosphate group. And so this was what makes RNA so unstable, right? It's very, very unstable. It's not good for long-term storage of genetic material. Now, it has better uses, right? Think about what it's used for. It's used in transcription translation, right? Think about what happens when a cell is under one condition. So under a certain set of conditions, you want to transcribe a certain set of genes and turn that mRNA into protein, right? Now, what if those conditions change? And they change all the time. You want the cell to be able to respond quickly. And one way that we can do that is to make sure that those mRNA molecules don't hang out for a long time. We don't want them to. We want them to become quickly degraded so that when the environmental conditions change and we change the transcription of our certain genes, we stop making those other proteins or we start making some other proteins, right? And so you want a tight coupling between gene expression and protein synthesis. Otherwise, you're making proteins that you don't need or could actually be harmful. And so it's a good thing that RNA is unstable in that way, but it's not good if you wanted to use it as a long-term storage for genetic material. The other reason why it's not good to have RNA as your material is because what's kind of interesting is that cytosine can actually undergo a spontaneous deamination in order to make uracil. So when you look at these molecules, it's very dangerous if this happens and it doesn't get repaired because, so if you look at our original strand, right, we have our cytosine here. And spontaneously that cytosine is deaminated to generate uracil. Now, if that uracil gets left there and you allow DNA replication to continue, right? You're going to make a copy of both strands of DNA. You're going to make new copies of the DNA, right? And so in our bottom copy, we're going to put a C on the other side and it's going to be just fine, right? But what happens when we see a U? What's the cell going to do? Well, the cell is going to pair that with an A and then fix that U and change that U into a T. So now we have a mutation of a cysteine to a thymine. And so that's a mutation. So what the cell does is it looks for uracil inside of the genome. And if it sees uracil inside of the genome, it's going to remove it. So if we use thymine instead of uracil in DNA, it's a safety mechanism so that we don't have all that deamination leading to mutation. So this is why thymine is better. Thymine better than uracil, right? No mutations. Yay, no mutations. All right, so let's talk about this three-dimensional structure of RNA, right? So one example of RNA that has catalytic activity is ribonuclease P, RNase P. And RNase P cleaves nucleic acids, and it's really cool because this RNA folds back in on itself and makes a complex double-stranded helices inside of the RNase P molecule. And so because of all this overlapping and complementary double-strandedness, it leads to increased stability. So you have increased stability, you prevent autocleavage that we talked about because that OH is protected, right? And so when we look at this, what we're really looking at here is the two-dimensional structure. We actually haven't gotten to the three-dimensional. We'll talk about three-dimensional in a second. But when we talk about our two-dimensional, we have a duplex, right? It's a single strand, but it's folding in on itself in order to interact with itself. And so you can make all these different two-dimensional secondary structures. So here's an example of a plain old duplex where we have that base pairing between residues on the same strand of RNA. You can also have places where you have a base that doesn't pair. We call that a bulge, so that uracil right there is a bulge. You can have a hairpin. So a hairpin is you have to start with a duplex, and then that duplex ends in basically a big bulge. But it really looks like a hairpin, right? If you've ever used hairpins before, ladies, you basically have two, and then you have the loop, right? So that's your hairpin. You can also have a loop. A loop is like a hairpin, except you don't have a duplex that it ends in. So if you look here, here's our loop, right? We have duplex, duplex, then we have a loop. So it's like a big bulge. And then we have more duplex, duplex. So a loop is kind of a combination between a hairpin, well, between a bulge and a duplex sort of deal. Okay. All right. Let's see. So now let's talk about three-dimensional structure. When we talk about three-dimensional structure, tRNA is really the one that stands out. And in order for tRNA to work, it has to maintain a particular tertiary structure. And so what you see is really the secondary structures that we talked about before. But these dashed lines here, these are places where distant places now come closer together and interact. And so you go from almost like a plus sign structure to now an L structure. And so those dashed lines are those three-dimensional connections or tertiary folds where you have secondary structures interacting with one another. And so this leads to a lot of stabilization, right? And you need all these folds really because the tRNA has to interact with the ribosome, has to interact with the enzymes, right? And the reason that this leads to stability is because you have stacking You have stacking properties. Hold on, there's Charles. Hold on. What you need, buddy?
Can I have ice cream?
Yeah, you can have ice cream. What? What? Okay, so I think we were talking about why the three-dimensional structure leads to stability, right? So we have stacking of the helices, right? We have loop interactions, right? And you can also have modified RNA bases, and we really haven't talked about that yet. Modified RNA bases, right? And so here are some examples of the modified bases that we're going to find in RNA. And a lot of times it's the addition of a methyl group in place of the hydrogen on the former hydrogen bond, in place of the hydrogen on former hydrogen bonding nitrogen donors, right? So normally that nitrogen is going to be a donor, but instead of having that hydrogen there, we're going to put a methyl group there. And so in essence, what you're doing is you're removing hydrogen bonds. And so they prevent certain structural configurations, and at the same time, it might facilitate others. So it's a way to regulate the tertiary structure of RNA molecules, whether or not you methylate or not. And so a lot of times addition of this methyl group, it's a hydrophobic methyl group, will actually contribute to base stacking. So it'll help those base interactions within RNA, but not always. But you can also have a functional role. So think about translation, right? If you have translation, in translation, a tRNA anticodon is going to have to match up with each codon in mRNA, right? And add the correct amino acid to our protein chain. But we have tRNAs that have anticodons with the nucleotide inositine, and this is a base and a sugar. And this base can pair with cytosine, it can pair with uridine, it can pair with adenosine, guanosine, oh, not guanosine, take it back, not guanosine, with these three, okay? 1, 2, 3, all right. And so what this means is that one tRNA can encode, encode, right? 3 different codons, right? So we get the same amino acids from 3 different codons. And I just realized my picture's kind of all over that. Okay, so let's take an example. What if you had GA, and instead of our normal, you're gonna have inosine, okay? So this is our tRNA anticodon. What are our 3 possibilities, right? Well, it could be CUA, right? This could be our mRNA. What else could it be? It could also be, gotta be the C and gotta be the U, but it could be uracil at the end, right? Or it could be CUC. So we're looking at this last codon, this last nucleotide in the codon can vary, and we're still going to get the same amino acid, and this would be leucine, regardless of what our mRNA was in that 3rd position. And that's why you have to have that codon chart, if you've used that before. And they came out with a new codon chart, which is a wheel, which I think is actually a lot easier to understand if you've ever used it. I don't know if y'all have used it before. You'll have to tell me in class. Okay, so we're going to talk about some other unconventional base pairing in DNA and RNA. And you can have an interaction of 3 strands of DNA, 3 strands of RNA, RNA and DNA, right? You can have lots of really different, interesting combinations, and you can even get quad where you have 4, right? And it occurs actually in a lot of guanine-rich regions. And it doesn't happen very often, but they can have functional and structural roles in the cell. Like you're going to form these things when you're trying to regulate transcription, right? When you're trying to maybe block a site, or it's a structure that's required for RNA processing, or it's a structure that's required for the assembly of a nucleosome. Sometimes it's in DNA repair. DNA repair is some really interesting things. The quadruplex, this one is typically only for transcriptional regulation, but our triplet has quite a few other purposes. All right, so how much does the DNA interact with proteins? And the answer to that is sometimes it's very specific and sometimes it's not very specific at all. And we talked about non-specific already. We talked about how the histones, for example, are positively charged and they interact with our negatively charged DNA backbone, and the DNA can wrap around that nucleosome and stabilize it and compact it, right? What we're going to talk about is specific side chain interactions of a protein with specific DNA sequences. And the way that this is done is through optimization of hydrogen bonding. Okay, so, and that's the end of, that's my end of 3.1, right? And we're going to talk a lot more about how these proteins here specifically interact with the DNA when we talk about restriction enzymes and when we talk about promoter regions and things like that. And we'll get into that in a lot more detail. But that is the end of 3.1.